Hi, I am Harish from KTH Royal Institute of Technology, Stockholm, Sweden. I'm giving my talk on fiber-based optofluidic microflow cytometer collecting side scatter light. This work was done at Department of Applied Physics at KTH in collaboration with Research Institute of Sweden, that is RISE, and Science for Life Laboratories, that is SciLife Lab. Flow cytometry is an important part of the medical lab. It's a gold standard technique for rapid detection and counting of cellular populations. Flow cytometry is routinely used in basic research, clinical practice, and clinical trials. And some of the uses of flow cytometry are cell counting, cell sorting, determining cell characteristics and function, detecting microorganisms, biomarker detection, protein engineering detection, and diagnosis of health disorders such as blood cancer. A standard flow cytometer looks like this, where there are several particles mixed in a sheath that are forced through an orifice, and a laser light shines light on these particles, and the light is scattered. If these particles are fluorescently tagged, they also scatter fluorescent light, and then the front and the side scattered light is collected using several detectors, and if the light is of different wavelength, they are filtered through dichroic mirrors and filters, and then they are analyzed to find several properties of these particles. In this work, we are trying to make fiber-based cytometer with silica capillaries and optical fibers. There are several advantages of using fibers and capillaries for building a cytometer. The fibers already have micrometer-sized cross-section. They are low cost and they can be made in kilometers. Microstructured optical fibers facilitate integration of light and liquid. Optical fibers and silica capillaries can be assembled to build optofluidic components, and these optofluidic components can be made using telecom instruments. These are some of the examples of the fibers that are available to us. The first diagram here shows fiber with 250 micron outer diameter with 5 holes inside of 50 micron each. And the figure B shows a silica capillary with the inner diameter of 250 micron and outer diameter of 330 micron. One more capillary here with the inner diameter of 125 micron and outer diameter of 250 micron. We can also have silica capillaries that are standard telecom size fibers with the 125 outer diameter and 90 micron inner diameter. These fibers can be spliced or manipulated otherwise using a glass processing station called Vitron, and we build the optical fiber components with these fibers. For the fiber-based microflow cytometer to work, we need a method to streamline the particles inside these silica capillaries. For this, we use a well-known technique from elasto-inertial microfluids, where we mix the particles in a viscoelastic fluid, and then we force this fluid through the silica capillaries. Due to the properties of the viscoelastic fluid, the particles are focused at the center of the silica capillary, and then we can shine the laser on them to collect the scattered light. This slide shows the previous work which was done in our group. Here we have, this is a fiber-based microflow cytometer for counting. In this case, we have particles mixed in a viscoelastic fluid coming in from this end. They are streamlined and focused at the center of this capillary. And in front of this capillary, which is aligned to this capillary, is a double-clad fiber with light coming from the inner core. So this light shines light on these particles one by one, and the scattered light is collected back in the outer clad of this capillary. In this work, we are only collecting the back scattered light, and there is no side scattered light collected because this is a dummy fiber used to just align the fibers together. So for a standard cytometer, we have to collect the forward and side scattered light. So in the present talk, we are trying to collect the back scattered and the side scattered light in order to perform cytometry on these particles. The schematic diagram of the flow cytometer we have developed is given in this diagram. The particles that are focused in a silica capillary due to the viscoelastic nature of the fluid, are entering the silica capillary from this end, and the laser light is incident on these particles. The light is scattered in all directions. The light scattered in the backward direction is collected by the outer core of the double clad fiber, and the light that is scattered to the sides is collected by another fiber which is cleaved at an angle of 45 degrees, and the end tip is coated with metal. The cross section of the silica capillary is given in this diagram here. Here it has an inner diameter of 90 microns and an outer diameter of 125 microns. And the angled fiber is prepared in order to collect the side scattered light. Now we are going to see how to prepare this angle cleave fiber. In order to collect side scattered light, we cleave a multimode fiber at an angle of 45 degrees using a dicing machine, and then we coat the tip of this multimode fiber with aluminum. The thickness of the aluminum coating is 50 microns. This angle reflects light coming from the side scattered light, and then it is coupled into the core of the multimode fiber. This picture shows the end face of this metal coated fiber. Once we did this, we coupled red light into this multimode fiber, and in this picture we can see that the light which is coupled is coming out at 90 degrees to the fiber. Next, we fabricate the component that we use for microflow cytometry. We insert the angle cleave fiber and the silica micron capillary inside a housing capillary, which is shown here. In this case, with the outer diameter of 330 microns and inner diameter of 250 microns. The full component looks like this, with the 90 micron capillary aligned with the angle cut fiber and the double clad fiber aligned with the 90 micron capillary. In this case, the particles will come in this direction. The light is shown from the DCF fiber, and the scattered light is collected in the angle cut fiber, and the back scattered light is collected from the double clad outer cladding of the double clad fiber. This is the experimental setup. We have a 450 nanometer laser diode with emitting blue light, and it is coming into this fiber optic component which we have fabricated. And we also have a microfluidic pump that contains viscoelastic fluid. The viscoelastic fluid has three different size of beads, that is 5 micron, 10 micron, and 15 micron in a viscoelastic fluid, and it is pumped through this microfluidic pump, and the particles flow through this component. The particles flow through this component and is let out at the fluid out. The side scattered light is collected and split into the fluorescent and scattered light using a dichroic mirror, and then it is aligned in a PC. This slide shows some of the results which we obtained. In this case, the flow rate with the microfluidic pump was 20 microliter per minute. We did 3 trials. In this case, the first plot shows the time trace of the results, and we took histogram of these time traces. In this, we can see only a single peak in the histogram, and it doesn't reflect the particle sizes in the viscoelastic fluid. Next, we increased the flow rate to 30 microliter per minute. In this case also, we did 3 trials, and the time traces are seen in this diagram. And then we took the histogram of the back scattered light, which in this we can see 3 distinct peaks, which is here, here, and here. The third peak is very not discernible. And the side scattered light also had also shows 2 peaks, which is given here. It doesn't show any other peak, but when we do a dot plot using the backscatter and side scatter light, we can see 3 blobs of data here, here, and here, which shows that the mixture had 3 different size particles. So we did for 3 times, and still we see similar kind of results. This shows our fiber-based microflow cytometer is working. We also did the same experiment for different flow rates, and for 40 microliter per minute, we again got data that is not reflecting the size of the particles in the mixture. In conclusion, we were able to demonstrate a microflow cytometer using fiber and fiber capillaries. This can open a lot of possibilities for point of care applications. Thank you.